Friends, today we are going to start, or we are started as the chapter 5th principle of inheritance and variation. Today, in our previous classes, we have studied about inheritance and variation, and we have seen what are the impacts of that. Now, in this slide, we will be learning about the laws of inheritance. As Gregor John Mendel, who is the father of genetics, worked on a pea plant and come up with the three laws of inheritance. Those laws are principle of dominance, or we can say law of dominance. Second, principle of segregation or purity of gametes. Is also in a place of principle we can use law of segregation or purity of gametes. A third, principle of independent assortment. Okay, these are the laws. The law itself explained with the help of the word used into it. Let the first law be law of dominance. So what is the law of dominance deals? Just look into the figure. We have two plants. One is tall, one is dwarf. For tall we have written capital T, T. As characters are in pair, as chromosomes exhibits in pair, so we have written 2T. That is capital T. For dwarf we have written small t. When both get mixed, then the new species is formed, which is having genetically it is having capital T and a small t. You can easily see. And the plant is looking tall. Okay. If this generation is more again crossed, we can say self-pollinated and crossed, then the F2 generation we will find 3 are tall and 1 is dwarf. If we go to the genotype of it, the genetic structure of it, then we find the first 3 plants are having capital T. One is having pure capital T and rest 2 are having capital T and small t, capital T and small t. And the last one is having only small t, t. That means small t stands for dwarf and capital T stands for tall. So wherever the genome capital T is there, the plant is looking tall. So basically what happens? Capital letter is exhibiting the dominant character. Hence, if the dominant character is there, then the plant will be dominant. So this is the law of dominance. This you can see in this figure as well too. Here the cross is given between smooth seed and wrinkled seed. So in this it can be easily seen smooth variety and wrinkled variety both are mixed together to form the same type of genome structure or genotype or phenotype which you have studied in the previous diagram. Now coming to these flowers, one is having axial flower, one is having terminal flowers. When they are crossbreed, then the dominant character is shown in the terminal flower, then it is again crossbreed. So the same thing means if the dominant character is there, then the species will be showing the dominant set of character. Now coming to the next figure, this will make you understand the law of purity of gametes, or we can say segregation, law of segregation and the purity of gametes. In this we will find the parent capital A and recessive gene is small a. Then they are fertilized, the gamete formation is there, then they form A and A, having capital A and small a, the F1 generation. Then that F1 generation is self-pollinated and they form gamete, and male gamete capital A small a and capital A small a. So this is when the gamete formation is there, they are segregated into a individual genome. When individual genome is there, that means apart from being together in a pair, they do not get mixed. They have not lost their purity. Keeping them together, they maintain their identity or their characteristics or traits. This is known as law of segregation and law or purity of gametes. Okay, now this you can understand the same thing when we have a different same gametes. Okay, so genotype is given here, and this is known as checkerboard of Punnett square for F2 generation. This you have to remember how you will write the characteristics. Now this is the characteristics for x and x, that Punnett square which we have studied. This is a in this we will be using how to make a cross between an offspring, whatever offspring is having characteristics plus the dominant character. If the dominant character is there and a recessive characteristics there, so if the offspring is being crossed with the dominant character, then what will be the ratio of the offspring formed, and if the offspring is being crossed with the recessive character, what will be the ratio of offspring formed. So if offspring is being crossed with the dominant character, there are chances that every species will be having dominant traits. Why? When offspring is being crossed with a recessive character, then there are chances 50-50 chances that one can be a dominant and one can be a recessive one. So coming to the law of independent assortment, law of independent assortment deals with that the chromosome, when they are kept independent or whatever the circumstance it may be, the traits will going to express itself in its purest form, whether it is in contact with other characteristics or not. Whenever they get a proper time, they will show their characteristics. Now in front of you, now you have a dihybrid cross. In this we have taken two characters, round and yellow and round and green. This is a checkerboard. You must learn this, how to prepare this checkerboard. Okay, in this the F1 generation is there, round and yellow, when crossed to form a round and yellow, capital R small r, capital Y small y. After that the ratio is being checkerboard is there for dihybrid cross, in which you have to learn the sequence, how to write and then cross the and form the output what is going to have. There are two ratios, phenotypic and genotypic. Phenotypic ratio is a physical ratio, that is 9:3:1, and genotypic ratio you have to calculate according to the gene. So this is all for the day. Thank you very much. To prepare the notes. Okay, thank you.